Thank you again. My name is Vahideh. I'm working as a Ph.D. student at WINLAB, Rutgers University. WINLAB, which stands for Wireless Information Network Laboratory, is a research center at Rutgers University, and the main research area lie in the areas of future mobile internet, wireless and network testbeds, healthcare, IT, wireless security and privacy in communications. And our group, which is led by Professor Wade, is working at the intersection of communication and biology. And today, I'm going to talk about a new approach, our approach to drug combination for cancer and other diseases. Now, let me start with a well-known example, which is migraines and headaches. When you have a headache, you may drink a cup of coffee or take a Tylenol and aspirin, or you may take the combination of these three, which is available as a cocktail drug, but at a lower dose of each individual drugs, which is shown this combination is more efficient than that's taking single drug. So it's a kind of everything about teamwork here. But how can we find this opportunity to gain more with using less dosage of each drug? To explain this, let's look at this graph, which is the response of a drug versus its dosage. As we can see, the effective response of a drug increases with dose. And here we achieve the maximum effective response. But after this point, when we pass this point, the effective response still is rising, but at decreasing rate. So there is no much more gain if we increase the dosage of drug here. On the other hand, if we increase the administration of drug, if we increase the dosage, we also increase the adverse effect of the drug. So we are interested in a range of dosage that gives us the most benefit but least adverse effect. So here there is a trade-off. We can see here in the other graph that we are interested in the optimal dosage at which we get the best combination of benefit and harm, or we get best benefit and least harm or adverse effect. And also, you can see that by drug combination, we can lower the required dosage of each drug, so we can reduce the adverse effect as well. But in the case of migraine example, we saw that multiple drugs working together can have a lot of benefit. There are other examples such as infective diseases, HIV, or cancer, that combination therapy has been shown that is much more effective than using single drug. But why is that? How can different drugs benefit each other? Cancer and many other diseases involve many different factors. And if we want to treat this disease, we should hit all these fronts. So we need to combine all these drugs to tackle the disease, to hit the disease. And this concept can be considered from different approaches. For example, we know that there are multiple pathways involved in cancer. So, and every drug reacts with its target through its pathway. So in order to tackle all these multiple pathways, we need to combine all these drugs. The other approach here is considering this fact that a tumor consists of a genetically diverse population of cells, and a single drug might only be effective against only one portion of these cells. So a combination of multiple drugs here could be helpful and could be a hope to cure the disease. Now we need to find, we want to find this combination, and we need to do it in a right way. So we need to have right tools to do it. First, we should know how to combining drugs interact with each other. Okay, two drugs could be non-interactive. That means like adding one drug to itself. Or if you combine two drugs and the response of the combination is more effective than each individual, they are synergistic. And in contrast, if you combine two drugs and their response is less effective, so it would be antagonism. And there is a well-known formula equation for this that explain these concepts in terms of dosage. This is Isobel equation. And equivalently here is Isobel graph that we can see from this graph, this dashed line is for non-interactive case. If two drugs have synergy with each other, then we need lower dosage of each individual. But if they are antagonism, we need higher dosage. But the fact here is that if we want to test all the possible combinations of multiple drugs in laboratories, you know better than me that a vast amount of resources and time is required to do all these tests. So here, mathematical modeling can be very helpful to study the interactive behavior of drugs before going to test them. And so we can say that we need mass simulation and optimization tools to do that. Now, the question is that how we determine the optimal dosage in combination therapy. Here we have pharmacological specification of each drug as the input of our problem. We want to maximize the effective response of the drug. We want to minimize the adverse effect, lowering the required dosage, reducing resistance, drug resistance, or any other thing like reducing the cost. And our output is optimal dosage of drugs in the combination. So we have a general optimization problem here, which we want to find the dosage such that maximize the effective response and minimize the toxic effect or adverse effect. To solve this optimization problem, now we need mathematical modeling that represents these equations. I mean the equations that represent effective response and toxic response. There are different models in applied pharmacology. For instance, these well-known median effect equations that are general form of Hill equations. And also there are a few equations or modeling for modeling drug combination, such as, again, well-known Loew-based modeling and Bliss-independent modeling. But we should know when to use what model for which scenario. And more importantly, The thing is that these conventional black box methodologies do not reflect the dynamical behavior of the diseases network. So in order to provide a precise investigation of drug interaction in the combination, I mean the interaction between different agents in the combination or the interaction between drug and its target, we need a network approach. So for this reason, in this slide, I'm going to overview one of my recent work in this area, which is going to avoid mechanism-based toxicity in signaling pathway by optimizing drug combination. Here, suppose that we want to tackle this protein in this target pathway using inhibitor I1. But there could be another pathway in normal cell with a receptor for the same inhibitor. So in this case, my goal is tackling this target pathway and this protein, but also a healthy pathway in a normal cell is also tackled unintentionally, as we can see in this graph, that both protein has tackled. But now the question is that, what's the solution? One solution is in the combination. If there exists another inhibitor with less and different effect on the healthy pathway, then the optimal combination of two inhibitors, I1 and I2, could be found that blocks the targeted protein with less effect on healthy protein. So here we can see that these two pathways now respond to the combination of two inhibitors differently, as you can see in this graph. Now, our goal is to find the optimal combination, to find the optimal dosage for these two inhibitors that tackle target protein but not normal cell. You can see in this graph that we are interested in this sweet area. Here we get most blockage on the intended and target pathway and least impact on the healthy and normal cell. And this area could be considered as the optimal dosage for inhibitors I1 and I2. So considering all these factors, we can see that determining the proper dosage involving in a combination of multiple pharmacological agents is a complicated optimization problem. And our goal is solving this optimization problem and enhancing the potency, preventing the toxicity, lowering the required dosage, reducing the resistance. But the point here is that all these goals only achieved if there is a collaboration between different groups, the group of people who work in laboratory and provide pharmacological data from lab experiments, and the group who works on modeling and optimization. Then the resultant model should be validated again in this lab. And I hope that we can find this opportunity of this collaboration. Thank you.